So I'm going to show you how to read an academic peer-reviewed research article. The article I'm choosing to use was written by a former Palisades High School student, and she has now discovered um, a possible cure for HIV. So it's kind of cool. Um, and it's probably not something that is one of your topics. So it should be applicable, but not identical to what you need. So here's the article. First, you want to, of course, read the title, Robust and Persistent Reactivation of SIV and HIV by N803 and Depletion of CD8 plus cells. So that doesn't necessarily tell you a whole ton about it. It already has some confusing language in there, some terminology that will probably not be familiar to you. So what you want to do first thing is to read the abstract. The abstract will tell you if this is an article that you want to read further. And so what I did was I started highlighting the abstract. I highlighted in yellow the things that are important to read that I'm going to want to come back to, and then the green ones I highlighted with a different thing because I don't understand what they mean, right? And I want to make sure that I look up those words or try to figure that out at a later point in time. But here's how I read this. I'll look through and read it, and then if there's things that I don't understand, I will just put stuff or did a thing in their place so that I can understand what's going on here. So let me show you this an example. So human immunodeficiency virus, HIV, persists indefinitely in individuals with HIV who receive antiretroviral therapy owing to a reservoir of latently infected cells that contain replication-competent virus. I don't necessarily know what all those words mean, but if I reread that in a way that can help me to understand it, then it turns out better. So human immunodeficiency virus persists indefinitely in individuals with HIV who receive a treatment owing to a thing that contains a replication competent virus. You see, so now I understand that what I'm reading is something about a treatment and a thing, and I want to find out what those things are. So the next sentence goes on to this term latency, persistence, and reversal, which I'll have to figure out what that means. Interleukin-15 superagonist 803 in conjunction with the depletion of CD8 plus lymphocytes in ART-treated macaques infected with simian immunodeficiency virus. I don't know what that means, so I'm going to have to come back to that later. But as I read through, I find that it did not reactivate the virus production, which is something cool, and reactivation of the virus in vivo. I don't necessarily know what in vivo means, so I might have to look up that. And viremia, I don't know what that means, so I'll probably have to look that up. And then here's another one. Co-culture of CD8 plus T cells blocked the in vitro latency reversing effect of N803 on primary human CD4 plus T cells that were latently infected with HIV. I don't know what that means yet. But the conclusion of this abstract is the results advance our understanding of the mechanisms responsible for latency reversal and lentivirus reactivation during ART-suppressed infection. So that's what I read first. I read that, and then I determined that this is an article that I want to read and I want to learn more about. And so I highlighted the things that are telling me about the article and what I'm going to read, and then I highlighted the things that I'm going to have to figure out. And so later on, as I go through the article, I will make notes, and you can kind of see that those notes are down here because I've already done this. So then what I do is after reading that abstract, I then scroll down through the pictures and see if the pictures make any sense. And I'm looking at the pictures, I'm like, okay, these are clearly test results of something. They mean something, so maybe I'll come back to that, but it doesn't really look like anything that I can understand right now. And then I go down, there's more pictures, right? Also similar to the ones before. And then I go down to more pictures and I go down to the discussion because that's the end. So this one doesn't have a conclusion, it has discussion. So I'm going down to this part and I'm like, okay, what does this mean? So here is a description of, the thesis of the end of the discussion. So I'm looking at that, and then I notice this thing is an important word again. It pops up in here. I have to figure out what that means. And then here's describing what what is going on in this article. So I read to the end, and I'm like, okay, I got an idea that this study is defining an approach, a different approach to attacking HIV. And then this is what the further studies are going to do. So that's a good thing to help me figure out what they learned in the article, because further studies build upon what they learned in this article. And so now I'm going to go back. So check that out. And now I'm going back to this part here. So this is kind of the introduction that gives me some ideas about what they're talking about in the article. And so I look up some words. So viremia, I look that up. Oh, no, where is it? That's right here. So I looked it up, and it's a condition where virus particles circulate and reproduce in the bloodstream. Okay, so that makes sense. And then the immune-mediated clearing means that the virus is killed by the patient's immune system. Okay. Latency reversing agent. So there's no way to reactivate the virus once it's hiding. Okay. And then histone deacetylase is a class of enzymes that plays an important role in the regulation of gene expression. So gene expression with viruses, right, is a really important thing. And so this latency reversing agent, the thing that helps to reverse the virus hiding in cells, is specifically a histone deacetylase inhibitor. So it's working with gene expression, right? And then I look over here, and so the article is going to be talking about more potent or powerful approaches to reverse HIV hiding in cells, because currently, from reading this, we do not have a current way of fixing the latency. Okay, so HIV is killed by some sort of immune system assistance, and then it goes latent, and then we have no way of getting the virus away from being latent and the person's still infected. So then I keep going down to this part, and this describes for me in more detail about what's going on. So I learned here that CD8 lymphocytes, which are a type of white blood cell, contribute to the suppression of HIV. And then I learned that this is the hypothesis experiment, because it says on the basis of these observations we hypothesized. Okay, so they are going to deplete the CD8 plus lymphocytes, and then they're going to combine that with the latency reduction, and then they're going to try to enhance the virus production under this the art therapy, which is the what was art therapy? That was the antiretroviral therapy. Sorry, you always got to remember what the terms are, and then you got to scroll back up and find them. And so then here is what it tells you that they're using. N803 is the molecule that's used in this experiment. This little sentence here describes what the molecule is composed of, which if you really want to, you can try to understand that, or you can just describe it as the thing that's used, they're using for the treatment, because understanding what it actually is would be further research for you, right? And then they it says that this molecule is targeting the residual virus pool, so the latent virus, and then it's used to strengthen your immune system's response. And these are part of your immune system. T cells and natural killer cells, right? They are types of white blood cells, right? So that gives me a good understanding of what they did here. right? So they're using this molecule in this treatment, right? And then I can read through again, and it starts going into more details about the experiment. I don't necessarily need specific details about the experiment to understand what they were doing, right? But it's very important that they write about that, right? So then I go back to the end and I find out again that this therapy was really effective, right? And this IL-15 superagonist N803, that's describing the molecule that they used, right? The treatment. And so then what it did was this. It induced a highly robust and persistent reversal of the latency. And so that means that it scared out the virus that was hiding in cells, and then they used the immune therapy to then kill the virus. And so this is previously undescribed, so this is a novel approach. This is new research, and that's pretty cool. And then I go back to my conclusion again, and then it showed that it worked, right? And so that's pretty cool. So now I have an understanding of the article. So the article tells me they found a method, a therapy treatment, that was used to successfully treat macaques and humanized mice to to get rid of HIV, which is pretty amazing. And so now I have notes made in my article, right, that I can use to then summarize later, and I have a more full understanding of the article. Now, what I use to highlight this is that Kami extension for Google Chrome. So you can use that too if you want to. I put a link in the announcements. If you want to use a different one, or if you want to print it out and do this, you can absolutely do that. Whatever method works for you. I like this method because I get to, write in the article, and it saves it to my Google Drive, which is nice. Just click save, and it says it saved it. So nice. Okay. So that's an example of an article that was really, really difficult to read, but I managed to do it in under 15 minutes, right? I managed to figure out how it works. And so hopefully this is helpful for you. And if you have any questions, please feel free to ask.